The next way in B1 that they ask you to understand genetic information is by means of pedigree analysis. And the way they show this is with the following sort of diagram. They draw males as squares and they draw females as circles. So this is a male and female reproducing. And then quite often they show the offspring that they get. If you see here, this means that this couple have had a son, a daughter and another daughter. So parent 1, parent 2, offspring 3, 4 and 5. Now quite often it includes shading. So if I put a key here, if you're drawn like this, this could mean that you're normal, and then this could mean that you are a sufferer. So if we were talking about cystic fibrosis, okay, we could look at this. Now if I say that this is the exam question, you can see that parent 1 and 2 reproduced to produce 3 children, and one of their children was a sufferer, but neither parent was a sufferer. Now using your knowledge of genetics, that should tell you something about these two parents. Because of recessive illnesses, if your children have the illness, you must both have been carriers. So if we said that this was if we said this was cystic fibrosis, that would mean that this parent was heterozygous and this parent was heterozygous. Let me show you what I mean. If that's dad and that's mum as heterozygotes, the only combination of genes that you could have that would work is this pair. So if I do the Punnett square to show you, okay, half of dad's sperm would be normal, half of dad's sperm would carry the cystic fibrosis gene, half of mum's eggs would be normal, half of mum's eggs would carry the cystic fibrosis gene. So this child would be normal, this child would be a carrier, this child would be a carrier, and this child would be the sufferer. So this would be child 4, because they're the only one that's got cystic fibrosis. You couldn't have this combination of parents, i.e. one being normal and one being a carrier, because this is what happens then. Okay, let's do dad's sperm. All of dad's sperm would carry the normal gene. Half of mum's eggs normal, half of mum's eggs carry the cystic fibrosis gene. When they combine, all of the offspring have the normal gene, and because that's dominant, they would all be normal. Well, that hasn't happened in this case, because these two people have reproduced and produced a sufferer child. So normal with carrier can't be the case. And obviously, if you had normal with normal, there are no cystic fibrosis recessive genes, so they must logically also produce all normal children. The next question I might ask you is, what about child 3 and what about child 5? Well, child 3 could either be homozygous dominant normal or heterozygote and be a carrier. And the same is true for child 5. It could either be normal, homozygous dominant, or a carrier, heterozygous. Well, how does that work? Well, if mum and dad are both carriers, all of these children would look the same. You wouldn't be able to tell just by looking at them that they had a recessive illness. But because of that, you wouldn't know whether they were heterozygote or homozygote. And that's pretty much how these pedigree charts work. Quite simple, really. What you have to do, though, is remember these combinations and work them out. And it's to do with practice. And it's this thing again and again. This is the repeating pattern for monohybrid crosses. Just watch this. I'll do it very, very quickly. Okay, so. Okay, so what you would end up with is all of them carriers. And the next thing I want you to know is 2 carriers. And this is where you get the well-known ratios for monohybrid crosses. Oh, you silly man. That's rubbish. Okay. That's what you get for rushing. So this is your 3:1 ratio, normal to sufferer, and your 1:2:1 ratio, big F, big F, big F, little f, little f, little f. And it's those 3 patterns that you get all the time.